So kita tengok, kita mark sama-sama awak punya lab report. Mana? Okay. So the title is, what is the title of the experiment, class? What is the title of the experiment?
Cell division mitosis.
What? Two? Title? Cell division. Cell division, which is mitosis. No mark given, however it is compulsory as usual. And then for the objective, there should be two objectives. The first one is you have to prepare onion root tip slide. And then you need to identify the stages in mitosis. Remember, every time you write down objective, you should understand the objective, because you have to make conclusion. You have to draw conclusion based on the objective. So make sure awak bila baca, awak tahu objektif tu dan awak tahu how to answer. So kalau to prepare onion root tip slide, yang ni awak tak perlu pun. Bila dia punya result should be in action, contohnya to prepare. Awak tak perlu pun, ini tak perlu pun awak tulis dalam conclusion. Kalau tulis conclusion, awak akan kata is prepared. The onion root tip slide is prepared. Kita tak nak. Yang ni memang awak dah buat pun. So the second one it should be the conclusion. So macam mana nak jawab ni? Identify the stages in mitosis. Macam mana agak-agak conclusion awak? Saya belum lagi masuk yang lain, dah terus conclusion. Sebab kita tengah tengok objektif. So what is the conclusion, class? The stages in mitosis
are prophase, metaphase, anaphase, telophase.
Yes, prophase, metaphase, anaphase and telophase. Kan mudah je, dah dapat conclusion. Okay, and then for the method and procedure, it is 2 mark. 2 mark, however, you have to draw. Kali ni nampak tak, kalau eksperimen first dengan second tak silap saya, tak ada markah. Biasanya kita orang tak letak markah because it is actually, kita nak kata awak punya try and error. We are allowing some rooms for mistake kat sini. And then now kita dah start giving marks because you are expected to do better. So ada mark kat sini. Okay, ada 2 markah. Make sure awak punya 2 markah tu memang awak sendiri kena evaluate your own method and procedure. So is it deserve to receive 2 marks, 1 mark or only 0 mark? Kalau tak ada markah. Kalau biasanya 1 atau 2 lah, sebab kalau kosong tandanya awak tak buat langsung. Okay, and then for the drawing, saya dah pesan awak, you have to draw 4 plant cells under 4 different stages of mitosis. Okay. Kalau awak tengok kat sini, dia adalah mitosis. Remember, bila saya dah sebut plant cell, apa yang dimaksudkan dengan plant cell? Macam mana awak nak draw plant cell? Apa beza dengan animal cell?
Ada cell wall.
Cell wall.
Plant cell, awak kena make sure awak kena buat 2 membrane kat situ. There are 2 membranes. Another one is cell wall, another one is plasma membrane. And then make sure awak kena buat dalam bentuk kuat. Kita nak kata dalam bentuk rectangle. Dia biasanya rectangle in shape. Sebab apa? Kita ada cell wall. Cell wall give shape to the plant cell. Dia bukanlah macam animal cell. Animal cell basically dia bentuk dia tak ada fix. Dia bentuk macam bulat macam itulah. Okay, hati-hati bila tu case plant cell. Make sure. Saya rasa most of you ramai yang tersilap kat sini. Tak ada cell wall. Tambahkan cell wall awak tu. Make sure ada cell wall. Okay, so ada kat sini adalah 3 markah. Markah yang pertama adalah the correct drawing. And then the second mark given is for the label. Ingat ya, every time you draw, make sure you label. So awak label apa? You label cell wall, you label apa lagi? Ada cytoplasm, plasma membrane, kromosom. Yang paling penting adalah kromosom. Awak kena tunjukkan. Macam mana awak nak tahu the behavior of the chromosome? This is prophase. Awak boleh refer dalam buku SPM, boleh refer, saya tak ada masalah. Awak kalau nak lukis based on your tutorial book, lagi saya suka. Tutorial book adalah nak kata reference utama awak boleh tengok. Kalau awak cari internet pun tak ada masalah. As long as awak buat under, it is plant cell and then it is under prophase. So the third mark given is apa? Title. And then magnification. Okay, magnification awak nak buat apa? Yang ni. Okay, yang ni saya ada tak dalam ni? Dia tak bagi tahu berapa. Okay, so for magnification ni saya nak awak ambil ni under 40 times. Ambil 40 times. 40 times time 10. Maksudnya kat sini in total the actual magnification is 400 times. Sebab kita follow dalam step tadi tu. Awak kena letak magnification. Okay, then make sure profess macam mana the behavior of the chromosome. Nanti lepas ni awak kena, saya nak tanya what is the behavior of the chromosome. Macam mana chromosome punya drawing during prophase. And then metaphase sama juga 3 markah juga, tapi kat sini kita tengok the behavior of the chromosome. The behavior of the chromosome, make sure lupa saya nak bagi tahu berapa drawing untuk kromosom. Awak boleh lukis 4 or 3 kromosom. 4 ni maksimum lah, tak payah lukis sampai 10 kromosom. And make sure awak punya the number of kromosom consistent. Kalau prophase tadi awak buat 4 kromosom, remember metaphase, anaphase, telophase semua awak keep on drawing 4 kromosom. Kalau 3, kalau 2. Saya prefer untuk buat 2 lah paling minimum. And 4 is the maximum. Tak perlu buat 1 dan tak perlu buat banyak. Okay, and then for the metaphase, make sure all the chromosomes are centered or lined up at the metaphase plate. Metaphase plate ni basically kita akan buat putus-putus ni. This one is we consider as metaphase plate. Ataupun mungkin ada yang label as equator, boleh juga. Tapi kalau dalam matric kita selalu kita guna metaphase plate. Okay, equator pun boleh buat juga. Okay, then macam mana awak kena lukis? Awak kena make sure, okay macam mana nak lukis? Awak lukiskan dulu dia macam bentuk butterfly. Then awak kena, okay tengah-tengah ni adalah apa? What is the middle of the chromosome?
Centromere.
Centromere, yes, the centromere. So centromere ni, daripada centromere ni lah awak kena buat spindle fiber ataupun gentian gelendongan yang awak belajar masa sekolah dulu. Ada yang guna gentian gelendong. So this one is the spindle fiber. So it is centromere, they hold the spindle fiber actually. Okay, then kita proceed with the anaphase. Macam mana nak lukis anaphase ni? Saya memang kalau dalam kelas ni saya memang nak tengok seorang-seorang macam mana buat. Most of you ramai yang silap. Okay, tengok ya macam mana nak lukis adalah make sure you draw starting from the centromere, baru awak tarik keluar, Bukannya awak buat macam ni, salah kalau awak buat macam ni baru tarik, salah. It should be the centromere ni ditarik. It is actually the centromere being pulled to the pole ataupun ditarik ke kutub, pole of the cell. Dia akan ditarik kat sini. Awak kena buat bentuk macam butterfly, kata bentuk butterfly ataupun half of the butterfly ni, it is being pulled apart towards the pole of the cell, ditarik ke kutub sel. Itu dia. So kalau lukis macam bawah ni, this is wrong. This is not the correct drawing. Okay, yang betulnya atas ni, this one is the correct one. Okay, make sure you ada label, you ada title, you ada drawing and you ada magnification. Magnification awak tulis actual magnification berapa? Actual magnification is 400 times. Macam ni. Buat terus. Payah 40 times 10 tu sebenarnya tak complete lagi. Awak terus buat actual magnification. Itu baru complete. So telophase pun sama juga. Ada 3 markah. So telophase ni beza dia adalah macam mana nak tahu telophase dengan cytokinesis. Thing is, untuk tahu cytokinesis, it's not yet cytokinesis. Kalau kita tengok telophase, dalam telophase kita akan kata dia ada 2 nucleus. Now we've got one cell with 2 nuclei. Kita sebut nuclei lah sebab ada 2 nucleus, actually nuclei. So there are 2 nuclei in one cell. After the formation of 2 nuclei, then baru kita proceed dengan cytokinesis, which is the splitting of cytoplasm at the middle. Tapi untuk plant cell cara dia kita akan guna istilah apa ya? the formation of apa namanya? Something plate. Alah, saya dah tak ingat dah. Tapi tak apa. Bukan penting untuk dalam eksperimen kali ni. Apa namanya? Saya dah tak ingat dah. Tapi plate lah something. Kalau dalam animal cell, it is called cleavage furrow. Cleavage furrow.
Cell plate, Miss.
Yeah, very good. Cell plate. Betul. Cell plate is in plant cell, cleavage furrow in animal cell. So dua-dua ni adalah cytokinesis. Saya sangat suka topik ketiga ni, topik eksperimen ni dengan cell division. Topik ni sangat menarik. Okay, and then kita tengok discussion. Total tadi adalah 12 because of 3 times 4 drawing. Each of the drawing consists of 3 marks. So in total there will be 12 marks altogether. Okay, and then for the discussion, you have to discuss the event occur at each stage in mitosis. Ni awak nak refer kat mana? Refer lah dalam buku tutor awak, buku lemon awak. Awak ada kan buku lemon? Siapa yang tak ada physically, awak boleh refer via link yang kita orang ada bagi tu. So kat sini awak boleh open page berapa? Kita refer sama-sama lah. The same page. Page
56.
56, very good, yes. Page 56. So kita tengok event of each stage. Semua orang buka ya buku nota ni. Okay, awak boleh tengok the first one is prophase. What is the chromosomal behavior? Chromatin condense. What is the difference between chromatin and chromosome and DNA? Apa beza dia pula tu? Okay, so kalau DNA, actual DNA macam biasa, 2 meter long, and then chromatin ni kita nak kata dia sebenarnya DNA yang DNA. Meaning that it is not yet shortened and condensed. Dia tak lagi menebal, tak memendek. It is actually long and thin, masih panjang. Okay? Masih panjang lagi. Okay, tapi kalau awak tengok ingat ya drawing dia, it is not macam ni. Drawing dia akan jadi ada 2. There will be 2. Ya, awak boleh nampak. Chromatin dia kat sini. Chromatin dia yang macam mana daripada atas ni jadi kat bawah? Macam mana? This one is, awak tengok this one is 1 DNA. Tapi awak tengok kat bawah ni kita ada 2 DNA. Why? Kenapa daripada 1 DNA become 2 DNA? Any reason? Awak dah belajar dah ni. Siapa boleh jawab? Because of?
Replication.
Very good, yes. Bangga saya. DNA replication. That's why you've got 2 DNA. Kan kita result DNA replication, we've got another copy of DNA which is exactly the same from the original DNA, from the parental DNA. So sekarang ni kita dapat 2 DNA. Yang mana kenapa kita dapat 2 DNA? Sebab nanti kita nak split tengah-tengah ni. Sebab nanti kita nak dapatkan 2 nucleus. So each of the nucleus should be getting the same exact copy from the original one. Then what will happen during prophase this one? Apa yang berlaku? Dia akan dia become shorten and condense. This one will shorten and condense. Chromatin shorten and condense forming chromosome. So kita akan dapat chromosome. Chromosome ni adalah result of shortening and condensing of chromatin. Okay, then apa lagi yang berlaku kat situ? Boleh tambah. The chromosome become visible. It can be observed, easily observed lah sebab dia become visible. Dia dah shorten and thicken. And then what happen? Nucleolus disappear. Nucleolus kat mana? Nucleolus is actually inside the nucleus. So this one is the cell and then we've got nucleus. Ini adalah animal cell, sorry. So this one is plant cell and awak boleh draw. This one is nucleus and then the inside one is actually nucleolus. So actually nucleolus ni disappear. Then what will happen? Nuclear envelope disintegrate. Kat mana nuclear envelope disintegrate? This one. Nak kata sekarang ni the nucleus dah mula tak ada. The envelope envelope dah tak ada. The last one is spindle fiber form. Spindle fiber, gentian gelendong tu. The formation of spindle fiber. Because ingat ya, the formation of spindle fiber begins when they want to start to divide the nucleus. Itulah function spindle fiber. Every time you see spindle fiber, you know that it will be in one of the stages of mitosis or meiosis. Dia akan bermula ada. Kalau sel tu tak ada spindle fiber, kita tahu the cell is not yet undergo mitosis or meiosis. Okay, dan kat situ awak dapat berapa markah? 2 markah sahaja. Awak tengok poin mana yang awak dapat. That will be 2 marks. So same goes metaphase, mudah saja. Metaphase ni macam mana? Okay. So metaphase ni dia kata actually paling mudah awak cari the chromosomes are apa? Apa? Sambung ayat saya. Metaphase.
Align at the metaphase plate.
Yes, line up at the ataupun line up or align at metaphase plate. Very good. And then what will happen to the chromosome and spindle fiber? Macam mana dia boleh line up? The centromere is attached to the spindle
fiber.
Siapa yang create spindle fiber? Which organelle?
Centriole.
Centriole, very good. Tapi kalau dalam plant, do plant have centriole? Plant cell has no centriole. However, there is spindle fiber. Ni sila cari jawapan kepada persoalan saya ni. Plant cell has no centrioles, but there is spindle fiber. How do plant cell produce spindle fiber if there is no centriole? Okay, cari jawapannya, nanti saya akan tanya dalam tutorial. Okay, and then what happened during anaphase? What happened during anaphase? Centromere split. Centromere split. So actually kalau tadi kita nak kata kalau sel ni bila dia dah split dia akan jadi ada 2 macam ni kan. So kita tengok ya. Ni sebenarnya basic awak ni. How many chromosome here? How many chromosome? One chromosome. How many DNA here? How many DNA here? Nampak tak? Saya nak semua orang try. Tak apa salah. Ini basic ni. Basic sangat ni. Kalau dah belajar kat sekolah, memang dah belajar pun. Okay, so how many DNA here?
2
DNA. One chromosome, 2 DNA. Now, kita tengok dia undergo anaphase. Remember, anaphase is the splitting of centromere. Splitting of centromere. Pegang ayat saya. Splitting of centromere. So now, how many chromosome? How many chromosome here?
2
chromosome. How many DNA? 2 DNA. Yes, ini jawapannya. Kalau tak faham lagi ni awak kena faham betul-betul ni. Kenapa from 1 chromosome become 2 chromosome? Sekarang ni awak tengok. Dia sebab dia dah separate, dia dah split, so dia akan ada 2 different individual. So sekarang ni kromosom ni dia dipanggil sister chromatic dan kromosom yang baru ni yang dah split ni dipanggil daughter chromosome. Daughter chromosome. Okay, itu istilah dia. Okay, dah. The last one is telophase. So telophase awak tengok balik dalam buku awak ni dia kata apa? Spindle fiber. Starting to mana? Telophase. Chromosome arrive at the opposite poles. Chromosome ataupun sister chromosome. Yang tadi saya kata sister, daughter chromosome, sorry. The daughter chromosome arrive at the opposite poles. Sampai dah dekat kutub tu. And then chromosome uncoil and lengthen, meaning that dia akan dia akan dia memanjang dan uncoil and lengthen. Saya susah sikit nak translate balik ni. So it will become chromatin again. So dia reverse process macam prophase. Senang je. Telophase dengan prophase dia adalah reverse process dia. Apa yang form, dia akan disappear. Okay, tadi kalau disappear, now dia akan form balik. Okay, so in total, 8 mark. Okay, dah nak beda?
Madam,
Then, yeah.
Madam, can we write the nuclear envelope has a nuclear membrane?
Oh, nuclear membrane, nuclear envelope. Boleh. Term dia. Tapi cuma nuclear envelope is more correct. Tapi nuclear membrane still understandable, boleh.
Okay, Madam.
Okay, then for the question and answer, few minutes left. So why is the root tips placed in the acetic alcohol? Kenapa? Because of we want to stabilize the structure and prevent chemical and structural changes during staining and mounting. Okay, so kita nak destabilize, so dia takkan rosak. So alkohol ni basically dia akan freeze. Kita nak freeze the chromosome. We want to freeze the nucleus there. So dia takkan undergo any more changes kat situ. Kita tak nak dia rosak, so basically acetic alcohol ni akan freeze. Dia akan stable the structure. And then what is the purpose of HCl tadi yang kita immerse for 5 minutes in 60 degrees Celsius HCl? Why? Because of we want to kill the cell. Oh, ayat dia. Basically we want to stop the mitosis. Kita nak no any more proceed. So kalau dia time to profess, dia takkan proceed. Through our experiment yang kita akan buat kan, kita akan undergo another step. So kat situ dia dah stop the process. So kalau kita tengok dia memang terus nampak profess. Mungkin nampak interface, kita boleh nampak yang lain juga. Then what is the most kita kata apa? Most observed stage of mitosis kat sini. Kenapa dia sebut mitosis? Dia bukan, soalan ni tak tepat. Kita tak boleh nak kata dia macam ni. Ni adalah cell cycle. Then kita akan ada interface. Interface ni lah sebenarnya paling lama sekali. It is about, okay, this one is cell cycle. 90% of cell cycle is actually interface. And then 10% nya adalah mitotic phase. So mitotic phase ni ada dua. So apa dia yang dua tu? Yang pertama adalah mitosis. Yang ada 4 tu dalam ni lah. And then another one is cytokinesis. Soalan ni first dah salah. Tapi soalan ni difahamlah apa yang paling banyak sebenarnya dilihat dalam cell cycle. It is interface the answer. Okay, then chromosomal behavior tadi awak dah jawab dah. Okay, so awak hanya salin balik what are the chromosomal behavior. Chromosome. It is about chromosome, not spindle fiber, not nucleolus. It is about only chromosome. Awak sebut chromatin, then chromosome, and then what happened to the chromosome, and then where does mitosis actively takes place in plant? Kat mana? Root tips and shoot tips. Root tips and shoot apex sama saja. Awak guna perkataan apex betul, awak guna perkataan tips pun betul. Dia sama saja. Okay, so berapa markah in total? In total question and answer ada 6 markah. Okay, so ada 5 soalan tetapi question number 4 consists of 2 marks. Okay, ada 2 markah. So 2 markah tu saya akan kata profess metaphase. Kalau betul baru 1 markah. Anaphase telophase betul 1 markah. Then conclusion kita dah buat dah sama-sama tadi. Apa dia? And then references. As